Alright, so we're just going to be doing a quick video on our gel electrophoresis processing that we had done. This is one of many times that we have done this process. So you can see here we had a preparation of samples and their proportions, and we had done 2 plant DNA samples and 2 animal DNA samples. We had also included a ladder as a control, and I just included a picture here where you can really see what the ladder looks like to kind of represent the bands in comparison to the samples. So for ours we had the ladder, and then there was 1 plant sample, 2 plant sample, and then farther down there was animal and animal, so that way they were able to be easily compared. You can see here where we had actually set our samples up. There is the animal DNA, the initial sample. They were prepared having the actual sample along with a dye, a loading dye, and those were moved into each of the tubes here. There's 1 sample for each, and then 1 sample for each. And then here's our ladder, and then we had also included an unknown sample that was provided. You can see here we are filling a buffer solution into the gel electrophoresis machine here. This is going to be filled in while the device is turned off, obviously to avoid any external current touching the body. You can see our samples here waiting to be loaded into the wells as well once they were properly done. And that was done using the smallest micropipette here to fill each of the wells individually. Following a gel electrophoresis at 90 volts, 95 volts for 45 minutes, you can see here the destaining process on the shaker, but the gels were actually soaked in a stain after being washed with water, and the gels are sitting within these containers here. So then they were destained using a shaker for a few hours following that. In terms of results, aside from a slight tear we had, you can't necessarily see it the best in this photo, but there's a little bit of a band here and here, as well as here there's a slightly darker color for the animal samples. When this was done, it should have been done with a different color dye, which we're experimenting with the following weeks, which will be in a later video. But even with the UV, it was pretty difficult to tell. Everything traveled in 1 band, and it seems like the sample has a low concentration of DNA in terms of the size of the DNA, because the bands were found way further down rather than being clumped up at the top. Although there were a few marks here and here as well that may indicate some DNA that didn't separate fully. So in conclusion, we had just kind of gone through a routine DNA gel electrophoresis process. We are trying with a new dye to get better results that are able to be seen visually, as well as going to try, obviously, to prevent any type of tears in the agar gel in the future. Thank you.